Hello aspirants. In this module, we are going to see what is extractables and leachables. So extractable leachables of late has become famous in the analytical laboratory setup because the Western countries have put some regulations on the quality of the product, whether it is meeting the expectations of the regulatory agencies with respect to the extractable and leachables identification as well as the quantification. So this is more or less related to the packaging materials used in the pharmaceutical products and whether the packaging materials having any interaction with the product and eventually it is forming some impurities in the product and the impurities whether they are toxic, mutagenic or not, and eventually it is going to affect the product quality as well as the patient's consumption. So once they consume such bad quality product, which is having the interaction with the packaging material, then the patient bound to get some kind of side effects or some kind of safety issues. So this is what being evaluated during the development of the product in terms of the extractable and leachables. So we are going to see what is extractable leachables in a brief manner in this particular module for understanding. Development of the pharmaceuticals brought a revolution in human health in last 70-80 years. And these pharmaceutical products would serve their intent or the application only if they are free from any impurities or leachables and safe to consume. So whatever pharmaceuticals we develop and market for the product consumption, it should be free of any kind of impurities or leachables and it should be very safe to consume. This is the basic requirement. So when you say the pharmaceutical product should be free of any impurities and it should be free of any kind of leachables, free of any kind of toxic materials, then you have to prove that the product is free of these kind of materials. How will you prove? By analyzing the product using various analytical techniques. So pharmaceutical analysis plays very important role to make the drug serve for their purpose, to analyze the impurities, leachables and other toxic materials from the product so that the product is free of these materials. So this analytical method will be developed at different stage of the pharmaceutical development, like stage 1 where we do the method development, and then the stage 2 we do the performance qualification verification, finally we do the validation and method verifications. We develop the method, then qualify the method, then finally we do the verification and validations. So when you talk about the pharmaceutical products which is free of these materials, then the analytical method also becomes very critical here to prove that the product is safe. When we talk about the impurities or leachables present in the pharmaceutical product, there are several incidents reported in the past with respect to the safety issues of the several products. For example, polycyclic aromatic hydrocarbons (PAHs) is a large group of organic compounds formed during the incomplete combustion of the organic materials. When you synthesize APIs, this is bound to happen. During 1990s, US FDA became aware of reports concerning polyaromatic hydrocarbons contamination within chlorofluorocarbon filled meter dose inhalers (MIDIs) and inadvertently introduced by some of the elastomers used as seals in MDI drug products. So this contamination they was notified by the FDA during 1990s. In some other case studies, the other reported impurity is the diethyl hexyl phthalate (DHP), which is used around the world as a plasticizer in PVC products. And this PVC is used for making intravenous tubing, bags, catheters, etc. And the regulatory agency found this DEHP in total parenteral nutrition fat emulsions, which is used for infants. And the DEHP is found to be very sensitive to DEHP triggered toxicity. So, based on these risks, in 2002, the FDA released a notification suggesting that alternatives to DEHP containing devices must be used in male neonates, pregnant women who are carrying male fetuses. So, this is one of the classical case of the leachables like DEHP present in the total parenteral nutrition products. So basically what we are trying to say here from this introduction that impurities and leachables toxic materials can present in the pharmaceutical products which is detrimental for safety of the product and it should be controlled. So this migration of mobile chemical species from the components used in the manufacture and storage of the pharmaceutical products must be assessed. So where from these impurities and leachables can be triggered in a particular product. So the impurities can be formed by the APIs interaction with the excipients or its interaction with the temperature or humidity, whatever reasons, the impurities can be formed. That is number one reason. Otherwise, the impurities can be formed from the migration of chemical species from the components used in the manufacturing parts as well as the storage of the devices of the pharmaceutical products. For example, you are filling the product in a plastic container or you are using a silicone tube during the manufacturing of the product. So some materials or chemical species can be migrating from the silicone tubes or the rubber stopper or the plastic bottles used in the pharmaceutical product for the storage or the manufacturing parts. So these chemical species can be contaminating the product. It can be the impurity and it can be toxic also. So this must be assessed during the development of the product. So that is called extractable and leachable studies. But what is the condition in India? As per the Indian Pharmacopoeia, the primary packages of pharmaceutical articles like parenterals mention visual inspection, clarity, transparency, leakage stage, pyrogen testing, and extractable metals. That is nothing but your elemental impurities. There is no detailed studies of extractable leachable required by the Indian Pharmacopoeia or Indian agencies, regulatory agency, but in case of US, Europe, Canada, and Australia, these leachable and extractables has to be performed during the development of the product and submitted along with the development report to the regulatory agency. And when it comes to the vaccines in India, leachable testings are performed but not for the other products. Here you can see some of the examples of guidelines which is talking about the assessment of and control of extractable leachables for pharmaceuticals and biologics. So this is essential part of pharmaceutical development nowadays for the Western countries including US, Canada, Europe and Australia. You can see another classical guidelines which is derived for extractable leachables by International Conference for Harmonization. This is the final chapter paper which is talking about the guideline for extractable and leachables, which is dated 30th June 2020. You can find in ICH Q3E. So it is talking about manufacturing or packaging system about a pharmaceutical product and how the extractable and leachable testing should be performed in particular product. So we are talking about extractables and leachables now frequently. So what is extractables, how it is defined, and what is leachables, how it is defined? Basically, extractables you will do using the packaging materials, and in case of leachable, you will perform the test using the product, finished product. So how will you define this? Extractables are defined as compounds that migrate from any product contact material when exposed to an appropriate solvent under exaggerated condition of the time and temperature. So basically you are testing the packaging material at exaggerated condition of time and temperature and identify the impurities formed from the packaging materials or the manufacturing parts. So those and all called extractables. What is leachables? The leachables may represent a subset of extractables that are found after the interaction between the product and the contact material under standard operating conditions. So the product is now manufactured and it is contact with the packaging material, and during manufacturing it was contact with the manufacturing parts also, and now you are finding some impurities in the product. So that and all can be coming out of the extractable list which we already established. So it can be a subset of the extractables and found in the finished product. So that is called leachables. So basically the extractables are intentionally established by subjecting these manufacturing parts and the packaging materials to a exaggerated condition, and you are profiling this extractable first, and then you are testing the finished product after aging or after particular time and checking that whether these extractables are present in the finished product. If it is present, then generally it is the subset of the extractables. So this is called leachables, what is present in the finished product. There are multiple sources which can contain leachable species, and some common examples are primary packaging materials, the external components like container closure systems like vials, caps, rubber stoppers, lids, foils, etc. Then the single-use systems like syringes and intravenous bags. And the internal components like gaskets, O-rings, valves, springs, etc. And sometimes the secondary and tertiary packaging, including the labels and adhesives and inks, also can provide these impurities and colorings. So these inks and adhesive can migrate into the product through the plastic materials. It is a possibility, a general possibility in the ophthalmic products. When you understand about extractable leachables, it is important to understand parallelly what is safety concern threshold and what is analytical evaluation threshold. So the safety concern threshold is nothing but the total daily intake of or total daily intake threshold below which a leachable presents negligible safety concerns from mutagenic and not mutagenic toxic effects. That means, for example, you define 5 microgram per day is your safety concern threshold, means below 5 microgram per day if you are getting the leachable, then you don't have any safety concern. If it is more than 5, then definitely you need to do the risk assessment. That is defined for different products by the regulatory agencies. So based on this safety concern threshold, you can calculate the analytical evaluation threshold, which is the concentration above which extractable and leachables need to be identified and quantified and reported. So the analytical evaluation threshold is nothing but the your lower limit of quantification for the analytical method, which is identifying the extractables and leachables, which is calculated based on the safety concern threshold. As I said in the earlier slide, the safety concern threshold is basically defined by the products, type of products. So the two factors which is defining safety concern threshold is nothing but the route of administration and the degree of product packaging contact. These are the two important parameters. So that's what overall we defined as a type of product. So based on the route of administration and the degree of product packaging contact, we have the FDA and the regulatory agency have assigned the risk of several products, type of products, and based on which the ACT is derived. So here you can see the table, the high risk, low risk and medium risk products based on the route of administration as well as the likelihood of packaging component and dosage form interaction. For example, you have a injection means vials and rubber stopper will be there. That is your packaging component, and the dosage form will be maybe a solution. So if it is a liquid solution and the packaging component rubber stopper and vial, then the route of administration is your parenteral, then it falls in the high category. The route of administration is also a high category, and the packaging component and dosage form interaction also falling in the high category. So it is high, so that you have to have a very stringent safety concern threshold for this particular dosage form. The highest will be the inhalation aerosols, where you have plenty of solvents, liquids you are using, and as well as the plastic materials. So it is the highest one, and injectable solution and suspension also falling in the highest one. And the lowest one will be the topical solutions, topical powders, oral powders, solid dosage form generally, oral tablets, etc., falling in the lowest one. So the highest one, that is the aerosols, will have a safety concern threshold of 0.15 microgram per day. And in case of injectable, it will be 10 times more, that is 1.5 microgram per day. And of late, regulatory agency recommended 5 microgram per day for the powder for suspension. That is the powder for suspension. It until you administer, it will be in the form of powder, and during administration, it will be reconstituted into the solvent. How will you calculate the analytical evaluation threshold? By using the safety concern threshold. So you have a product which is having 137 microgram per dose and the fill volume of 25 ml per bottle. So the dose will be 137 microgram per dose and the volume will be 25 ml in the particular bottle. So the strength is 137 microgram nasal spray, which is having 0.137 ml, and your maximum daily dose is 548 microgram, that is 0.548 ml. So the AET will be calculated using the formula of safety concern threshold multiplied with the dose per container divided by dose per day. That means your safety concern threshold for a nasal spray, which is the highest risk product, is 0.15 multiplied by dose per container. You have 25 ml, that is 25 divided by your maximum daily dose, that is 0.548, will give you the AET per container. Then this you will have the fill volume of 25 ml, so that you will divide it by 25, will get you the per ml concentration of the AET. So this 274 ppb is the AET calculated value, will be your limit of quantification for the analytical method to be used for the extractables and leachables. I have already explained that during development stage itself, we have to evaluate the extractable and leachables. So what are the analytical methods you are employ for a extractable leachable studies? And what are the target impurities or target compounds you are analyzing by using the different analytical methods? So the target compounds are nothing but non-volatile leachables, that is non-volatile organic compounds generally, then volatile organic compounds like solvents, etc., then semi-volatile organic compounds, leachables, then finally the element leachables. So basically it is divided into organic compounds and the elements, toxic elements and non-toxic elements, as well as the organic compounds. The organic compounds further divided into non-volatile, volatile and semi-volatile nature. So these compounds will be analyzed by different analytical techniques, like the GCMS is used to analyze volatile and semi-volatile organic compounds because it is volatile in nature. It will be mixed with the gas, and the gas chromatography will separate this compound easily and identify. Then non-volatile compounds will be analyzed by LCMS because you have to go for a low sensitive analytical method for these kind of impurities. Then the elements will be analyzed by using the ICP-MS method. As we have seen, there are several guidelines which is talking about extractable leachables assessment and how we have to carry out extractable leachable assessment for different kind of dosage forms. And here is the example of USP 1663 is talking about the assessment of extractables associated with pharmaceutical packaging and delivery system for different products. That includes the small volume parenterals where you can use the extraction solvents like water with different pH and isopropanol water, and in case of large volume parenteral, what you have to use. So this is some of the examples and extracted from the guidelines which is given for your understanding and information. So if you go to any regulatory document or a pharmacopoeial chapters, you can see more details about these particular procedures. What are the uses of these E&L assessments, the extractable leachable assessments? You can establish the worst case potential leachables profile in a manner which facilitates the safety evaluation and qualification of probable leachables. That is one use. Then the facilitate the assessment of patient exposure to chemical entities resulting from a packaging. So you can assess, one is the safety evaluation of the probable leachables, then you can facilitate the patient exposure to particular chemical entities from the obtained from the particular packaging, whether it is really safe or not. Then finally, you can facilitate the establishment of qualitative and quantitative leachables extractable correlations. So this also can be done. So you have done the extractable study and found the extractable profile, then you will do the leachable study and found some leachables, and you can do the correlation where from this leachable is coming, whether it is part of the extractable profiling what you have done already, and if it is found in the extractable profile, where from it came from the manufacturing part or it came from the packaging material, which part of the packaging material, whether it is really toxic or not, all those assessments you can do. So this is the basic use of these E&L assessments for the particular pharmaceutical product. Requirement of extractable leachable for the pharmaceutical products is essential for advanced regulated markets like USA, Canada, EU countries. And also we require, if there is a leachable found in the particular product, we need to do a toxicological or risk assessment for monitoring the particular leachable compound in the aged formulations. However, in Indian regulatory environment, as of now, there is no such requirement. In the previous slides, we have seen the importance of the extractive leachables and the various analytical techniques used for the assessment of extractive leachables, what is SCT, what is AET, etc. And now, whatever we have seen in the previous slides, we are going to summarize through this classical example of parenteral product. So here, I have selected a parenteral product for the development. So after the development or during the development, I am going to do the extractables and leachable studies for the regulatory submission. So based on the product maximum daily dose and the safety concern threshold, I will be calculating the analytical evaluation threshold and develop the method for various target compounds like volatile organic compounds, semi-volatile organic compounds, non-volatile organic compounds, and elements by using different analytical techniques. Once the method is ready, then I am going to first do the extractable study on the primary packaging materials as well as the manufacturing parts and created the extractable profile. Then I move on to develop the product, finished product, and manufactured it and stored for stability. So this stored finished product with the age of 6 months or 18 months or 12 months will be used for leachable study by using the same analytical method with the same AET and SET and see that whether you are finding any leachables or not. So if you find any leachables, you can correlate with the extractable profile. And here we can see the difference between the extractable and leachables. Generally, when you perform a extractable study, you will use extractable solvents in which you will find the extractables. So this solvent can be a placebo or it can be a exaggerated condition like isopropyl alcohol or water, or a pH, extreme pH like acidic pH or alkaline pH, in which you are artificially creating a extractable profile subjecting this product into exaggerated conditions like higher temperature and solvent system, and you are creating the extractable profile. Here in leachable, this is the finished product and you are assessing the leachables by using the same method. And if you are finding the leachables here in the finished product, you can correlate with the extractable profile. And in case if you find the leachable above the safety concern threshold, you have to do the risk assessment, toxicological assessment for the particular leachable. Whether it is safe or not, you have to establish. And in case if it is required to monitor the level of leachables in the products, you have to perform as part of the release of the product. So aspirants, in this particular module we have seen the basis of extractable leachables, why it is required and how it is generally performed, what are the critical aspects of these studies, and analytical techniques used, what are the target compounds, etc. Thank you.